Buenos días. En un minuto más comenzamos. Good morning and welcome to this seminary. The name of the seminary is Nitrate, Isotopes and Encycling in South Bohemian Lakes. Our speaker is Dr. Travis Medor. And we would like to thank the International Agency of Atomic Energy. And this project, this seminary is thanks to the project MEX 7014, and special thanks to Dr. Juan Pérez Quesadas for being in the gestion of this seminary. And without any delay, I'm gonna read some background of the speaker. And Travis received a PhD in chemical oceanography in 2008 from the Scripps Institution of Oceanography at University of California in San Diego, where he measured nitrogen isotopes to identify encycling and contributions by nitrogen-fixing cyanobacteria in the tropical Atlantic and Pacific Oceans. He then did his postdoctoral work at the Hellenic Center for Marine Research in Greece and Woods Hole Oceanographic Institution in USA, where he worked on the cycling of organic nitrogen and phosphorus in the ocean. He moved to the University of Bremen, Marum Center for Marine Environmental Science in Germany in 2010, where he used stable isotopes technique to measure microbial lipid production and carbon cycling in sediments and peatlands. He became a research associate in 2016 and led a project using carbono-14 and radiotracer techniques to measure the nutrient cycling by ammonia oxidizing archaea. In 2018, he began his current position as head of stable isotope facility at the Biology Center Czech Academy of Science, where he leads an organic biochemistry research group and collaborates on projects in the field of isotope hydrology and hydrochemistry, nitrogen and methane cycling in inland and coastal waters, plant and fish ecology, soil science, and ecosystem biology. And welcome, Doctor.
Thank you, Arthur, for the introduction. And yeah, thanks for everyone for your attention and showing up today. I'm happy. It's my first time in Mexico City, Ciudad de Mexico. And yeah, I'm excited to be here. And also that you're all interested in this kind of research. Yeah, so stable isotopes, as you heard, it bridges a lot of different fields of science. And I have a history of a lot of different applications of stable isotopes. And I haven't really focused on my interests on a single one of them, and my interests actually keep expanding because there's so much potential with this kind of research. So here's a photo just of our lab, and we have a mass spec back here, which is just one, and several peripherals. And so when you look at it, this is just a schematic of our lab, and so this is our IRMS, and then we have several components. And several of you know what this is, several not, but you can see that there's a lot of potential to measure a lot of different sample types. We measure solids, liquids, biomarkers, gases, and carbon, nitrogen, sulfur, oxygen, and hydrogen isotopes. And this is just a schematic showing briefly what is stable isotope measurement. Basically, it's a very, very sensitive scale, and so we measure the difference, or the ratio of weights of very small atomic differences in these CO2 molecule, for example. So we can tell the difference if something weighs 45 or 44 with very good precision. And that's the goal of the stable isotope measurement. So I'm going to present some of the ongoing research in our lab and the topics that we're working on, and one of the topics which brought me here, and then also give you some examples of how we study this in nature, so very natural cycles that aren't really influenced by humans, and then another example where there's a very strong human imprint on the nitrogen cycle. So, oh, I forgot. Yes, so we have a list of analyses that we do. So these are all the different kinds of things that we've measured over the last 4 years, and the different isotopes that we're measuring, and all the capabilities of our system. Yeah, so this is the research group. We have some students and technicians, and some technicians who are students, and basically we're trying to build the lab, but we have one instrument, so we can only accommodate so many projects. But the one that we've been working on the longest at this point is fungal lipids. So this is like fungi growing in soils, and we're developing stable isotope assay approaches to interrogate the fungi and ask them, what are you eating and how fast are you growing? And so we're hoping that this will take off. So this is the work of one of the PhD students who just had his first paper accepted. So we're happy about that. And I began working with groundwater and surface water isotope hydrology, and then given my background in organic geochemistry, I devised a way to combine these two, and so we're trying to create a new field where we're actually using the water isotopes in organic matter to define cycling. And so this is ongoing research and something I'm quite excited about. And and then in doing the isotope hydrology and groundwater, surface water, we started to get into nitrate contamination. And this was something that was also being pushed from the International Atomic Energy Agency, as they had developed a more simple method for that could be adopted in several labs. And so we were one of these labs that took their method and installed it in our lab. That had, we had to do some adaptations to get our system to work the right way, but in the end we passed the test. We got satisfactory results from one of their intercollaboration exercises, so that was a nice check mark. And then since then I've been going to different labs and suggesting how can they do the same. So and that's why I'm also here. So to identify what could be improved, or what is actually necessary to get the results, that you can have satisfactory data when you're measuring nitrates in certain types of water. And then we do a bunch of different collaborations, a lot with methane cycling and soil science, all kinds of ecology studies with plants and invertebrates and fish, and studying all different kinds of environments. And in my heart, I'm an oceanographer, so living in Czech Republic, that's been difficult, but I just got a proposal funded to go out to the Mediterranean Sea. So keep up, stick to your interests. It'll always come back. So now I'm going to talk more about the nitrogen cycling, and what we know from human impact on the environment, actually humans have impacted the nitrogen cycle far more than any other nutrient cycle on Earth. And this is a diagram showing that. So if you look at this graph is all reference to the input of human sources since 1950, which is the value of 1. And since then, if you look at the increase in CO2, it's not nearly as much as, it's more a threefold, whereas we have a fourfold increase in the amount of reactive nitrogen that humans have put into the environment. And this is coming mostly from fertilizers and this Haber-Bosch process. So if we look at the nitrogen cycle, there's definitely some natural inputs of nitrogen from the atmosphere that makes it reactive that we can use then for growing crops or primary production. And that's coming from nitrogen and microorganisms that fix nitrogen. And then since the development of synthetic fertilizers during the 50s, then you can see this is just ever increasing. And this demand has actually, this has been a demand that has allowed the human population to increase because we can make more food. And so this has been going on, for some centuries. And nitrogen is showing up in the fields and then flushing out into the waterways. And this is just an example. So of the inefficiency of our fertilizer and how it ends up actually in the consumption for humans. So if we start off with fertilizer in the field and then a lot of reactive processes that happen along the way. So this is the situation if you're a vegetarian. If it's a carnivores, it's much worse. So only 4% of the nitrogen that we're putting onto the fields is actually ending up for human consumption. So there's a lot of waste, and it's accumulating somewhere. So this is just a schematic showing in purple is the natural fluxes. And so this is, there's some kind of balance here, but since we've added some new sources of reactive nitrogen, this has created an imbalance. And if you add up everything that's going into the land and everything that's coming out, you'll see that there's more going in that's going out, which means the Earth has a way of balancing itself, and it means that there's going to be a greater flux of denitrification in the years to come because we have to get rid. You can't have this imbalance sustainable over long periods of time. So we have more coming in than is going out, and this is being transferred along waterways to the ocean. And so what I'm going to talk about now is a situation inland where we are looking mostly at a natural system. And then I'm going to look at another situation where we have more of the fertilizer and agricultural system. So these are two case studies that describe how we can use stable isotopes of nitrate to reveal these kind of processes or the effect of nitrogen loading in certain systems. And the first story will be about some pristine lakes in Czech Republic. So these are glacial lakes. Actually, there's not so many. Most of the lakes in Czech Republic are reservoirs, man-made reservoirs, and these were created in the last ice age. And so this is Čertovo and Plzeňa. And these are located right on the Czech-German border. And so we have a long history in our institute of sampling these lakes. They go up there every 3 weeks when it's not frozen and collect samples. And and so this is what they look like. So each lake has a couple of different tributaries feeding into it, and they have one outlet, and so there's a bunch of different sampling sites. They don't always get samples from all of the sites. It might be that one tributary is dry during the sampling season, but anyway, you'll see a couple of different plots that have different symbols, and that just means, these samples were taken from different area around these lakes. Okay, so the story about these lakes and why they started to get studied so much is because there was a lot of atmospheric nitrogen deposition. And this is coming in the form of acid rain. So when there was a lot of industrial burning and not clean burning of specifically like fossil fuels and coal, you can get a lot of nitrous oxides into the atmosphere that react. And they come down in the precipitation. So things that were bringing the precipitation into Czech Republic, we saw since the 1950s a strong increase in the amount of nitrate being deposited from the atmosphere into these lakes. And the lines before, I think, 1950 are mostly from modeling studies. And anyway, what you also see is this decrease in the pH over the same period of time. But a remarkable thing is that when you have this kind of observation and you have this scientists saying, informing the public, look, there's a lot of, there's acidification of our lakes, there's acid rain, this is the consequence, then there was some action. And actually the values started to decline and the pH started to recover. And so it just goes to show that, excuse me, with some data and conversation with policymakers, you know, things can improve. And so following this atmospheric deposition era, then there was other events, natural events that happen in the lake, and there's what's commonly reoccurring in this forest area is a infestation of a bark beetle. So it's killing all of the trees, and basically the trees are storing so much nitrogen from, you know, their biomass. When they all die, then you have a big loading of nitrogen into the system. And this is a more natural type of loading because this is nitrogen that's processed through all of the soil reactions, basically nitrification. So it's turning leaves, organic nitrogen, into nitrate. And then this is washing into the system. And so we saw big loadings of nitrate into these pristine lakes just from this everything dying off and leaching from the soils into the waterways. And not only do you have this contamination in the lakes, but then the soil is also drained of its nutrients, so it's hard to regenerate life. And so the situation was very drastic in the Plesna Lake, and in Čertovo not so much, but nowadays we're expecting that the bark beetle infestation is occurring in the other system now, and so there's going to be another pulse of nitrogen coming from here. And so, yeah, this is the current system. So now Plesna Lake is very, very low nitrate levels. We can hardly measure nitrate isotopes because they're quite low. And then this system is starting to pick up again. So we decided to, let me just go back. So we decided to investigate the nitrate isotopes in these two systems to learn more about nitrate isotopes as a diagnostic process for looking at the different inputs and also the health of the ecosystem. So if the trees are growing healthily, then what is the composition of nitrate in the lakes and what's accumulating, as opposed to the other scenario when they start to die off or they're not really producing much biomass, then we would see a different isotope signal. And to do that, we're going to measure nitrate isotopes. And this is just a brief review of how we as scientists have measured nitrate isotopes in the past. So it started with just strong reagents and chemical conversions, you know, first converting ammonia, nitrate to ammonia and then to N2 gas and measuring the nitrate, the isotope composition of the gas. And then eventually, I'll just get down to this box right here, which is the techniques that are most commonly used today. Actually, the most common method that I'm aware of for the labs or that I'm in communication with is this denitrifier method. So they use a living organism to convert nitrate to nitrous oxide. And the advantage of converting things to nitrous oxide is now you can get the isotope composition of not only the nitrogen, but also the oxygen. So it's telling more of the story of how nitrate is processed in the environment. And so this is one method that's been used. There was a chemical method also but this has some quite harsh reaction conditions, and the throughput is quite low. There's only so many samples you can process in a day. And so this was the motivation of Mark Altabet and Len Wassenaar from the IAEA to create a more easy method that could be installed in labs worldwide and have high throughput. And that's the use of titanium chloride to reduce nitrate to N2O. And so that's what we've installed in our lab, and that's and and um, and that's what that's the method we're using, and that we'll talk about today with the data that we have from the Czech lakes today. So this is just a schematic of the method. You add your sample, you dilute your sample to a known nitrate concentration. So it's very important going into the process that you know exactly how much nitrate is in your sample. You make sure it's under acidic conditions. Then you add this titanium reagent, you cap it and swirl it, and then the next day you measure the N2O. And so N2O has a mass of 44, 45, and 46 when you're detecting with isotopes. So it's the same as CO2. So it works really well with the cup systems that are already in a lot of the isotope machines. And it's quite straightforward. So at the IAEA, they use 40 milliliter reaction vessels, but the total volume is only 3 milliliters. So you could use smaller volumes, and that's what we do in our lab. And we'll talk about the specifics of this more with the team the isotope team, who probably has more interested in this. And then this is just a schematic of how we changed our gas bench in order to do these more trace level analyses of N2O in the headspace. And so, yeah, so it's flushing, you need a flush line, so you don't have to wait forever to get the sample into the mass spec, and you have to trap it with liquid nitrogen. And then there's all kinds of ports and things to clean up the sample and separate it before you send it to the IRMS. So this is just a schematic of that. Yeah, so these stars are just the innovations that we did to our system so that we could measure N2O reliably to get isotopes of nitrate. Yeah. Okay, so then we measure it, and then we get some data, and then now we have to interpret what does it mean. And over the years, this is a schematic from 2007 of the different basically boxes that are assigned that identify where nitrate, the sources of nitrates in different ecosystems. And so this is kind of used. So when we measure the nitrate and we know it falls into this box, and it's kind of more positive on the 15N, then we know it's coming from some kind of manure or septic wastewater treatment plant sort of systems. And this box is basically the natural samples. So if you just take nitrate that's coming from like poor water or where there had been nitrification by different organisms, and it usually falls here. Then if you're increasing in the 18O, it's coming from fertilizer. And then the atmospheric nitrogen has the most positive values. And this is because of reactions with UV in the atmosphere. And so the fertilizer that we're using it's coming from this Haber-Bosch process, which is taking atmospheric nitrogen and making nitrate. So it's a little bit more enriched in 18O, so it has more positive 18O values. And what you can see here is these boxes are quite big. And over the years, they're not getting more refined. What you have to do if you're going to study a system is figure out what is the box for the natural system, what is the type of manure that's being used, if it's poultry or if it's from different livestock, and what is the kind of fertilizer. So you have to define these end members for your system before you can go and make an assessment. And this is the data that we have from the Czech lakes. And on the left is Plzeň, on the right is Čertova. And so I've just marked the different kind of samples with either a X or an open symbol or a filled symbol. And the one takeaway I just want to take from the left plot is that we have, some background which is falling into this natural systems box, and then there's some that are enriched in 18O. And we know this is a natural system. There's no farms around. There's really only one explanation for how we can increase the 18O of this nitrate. I'll get to that in a minute. On the other system, we see this kind of trajectory over the course. So these are samples that were collected over the course of seasons and in different locations, and you can kind of make out that there's some kind of trajectory or regression line you could fit right here. And this is telling us that the biological activity of processing of nitrate is active. So one of the processes in the nitrogen cycle is this denitrification or removal of the nitrate from the system and back to nitrogen gas. And when you do that, you fractionate the nitrate basically, and then it increases in 15N and in 18O, and it looks like this. So the conclusion for this system would be like that the dominant process that's determining the nitrate isotopes is denitrification, which is happening mostly in the summer months, and it's kind of very it's identifying this natural kind of processing for this lake, which is quite different for this lake, which shows much more scatter and higher values. And if you remember the precipitation, if you collect the precipitation at the site, you can measure how much nitrogen is coming in from this avenue, and then also identify the isotope composition. And so now we have this end member from the precipitation and from the soil, and then you can use this to do kind of simple mass balance calculations. So the next slide, I think I only use the 18O values to try to ask the question, how much of atmospheric nitrogen is coming through the forest and into the lake? And basically, this is the hypothesis here. So this is described in the study, but you have this deposition. You can be processed here, so it goes into the vegetation dies, it is experienced nitrification, and then it's leaking into the stream. Or the deposition just goes straight into the stream, and so it kind of skips this whole forest cycle, and then that's the signal that we see. So, and then if we look over the course of against this, just the spring samples, you can see that somewhere up to 35% of the nitrogen that's coming after the snow melts and everything is derived from atmospheric sources. And whereas in the other system, it's really quite low, and all of the atmospheric nitrogen that's being deposited is still going into the vegetation. And so we think that when we have more data and a longer seasonal study, we can start to see the effects of forest health on nitrate isotope composition in the system. And this is the PhD student who's leading this. And so I think let me just make sure that's the last slide, right? Yeah, so that's the end of this story. And then now I'm going to switch gears and go straight into another study that was done actually by Len Wassenaar. And so this is a long research campaign for one of his sites that he studied, one of the aquifers that he studied in Canada. And this is just a paper. So if you want to read more about this, you can check out this paper. And it's this Abbotsford's Sumas Aquifer, which is located right on the border of US and Canada. So these international aquifers that expand different national boundaries, there's a great interest. And this one in particular, because well, there's not so many people there, but it's a water source. What is known is that there's a large contamination from all of the farms here. So it's mostly berry production, and the general flow path of water is from Canada to the U.S. So they're bringing all of this contaminated water across this international boundary, and so you can see how these residents in this state here are influenced from the agricultural practices in another country. And so it's been a great concern for several decades. And basically there was always poor management, once they started to get in fertilizers. So they were increasing the amount of fertilizer or cropped, the amount of crops being produced was increasing each decade. And so there was big interest to try to identify which best management practices need to be in place to curtail this kind of contamination. And so step one, again, was identifying these different end members. And so you have this box which shows the general composition of nitrate isotope composition, of nitrate isotopes. And so for this system, they went and they found that the poultry manure actually had very positive values. Sorry, these circles are a little bit off. And then if you look at the fertilizer, so this is the ammonia sulfate fertilizer, it has also a value around 0. So it's not this is the 15N value. And then if you look at the water, you can tell that the water is mostly influenced from this manure. So right away, it's very clear that that all of the nitrate that they were detecting in the system was coming from this manure. And so then they designed a bunch of laws and agricultural practices to reduce the amount of manure that was being leached from the fields into the groundwater. And so they installed all kinds of different best management practices, and then they looked at the situation a decade later. And so this was the original study in this blue, and then a decade later in green. And what you can see is from this plot is the earlier samples all plotted here with the manure, and then the newer ones plotted here, showing that a lot of the farmlands had switched from manure to the inorganic fertilizer, which wasn't regulated, right? So they could do all of the dripping techniques and everything. And so this and what I want to show, and also this plot, is you again see this kind of trajectory where they're looking at where you can see that denitrification is happening, which is nice if you have denitrification, because then it's a natural way to remove this contaminant from the system. But in the system, these were all samples that were from 30-meter or deeper wells, and so it takes a longer time for the water to get down there. And in these, when you only look at a decade later, it actually you're looking at more like surface water wells, something like 10-meter or 5-meter wells. Then it doesn't have time to be processed naturally. And so if you want to have best management practices for the water resource of the aquifer, you need to focus on things that will contribute to the removal of nitrogen or nitrate from the surface waters. And I just want to mention that because there's all kinds of processes going on, and so then they had to apply this helium, tritium dating to know which of the wells do they need to study in order to have a best management practice. So what I'm talking about is nitrate isotopes, but in the background, there's a lot more hydrology studies going on so that they can know where to sample and when to sample. And so so then they went back to do another study. Wait, so let's summarize this one more time. So I just wanted to talk more about the age of groundwater. So if you look at the older groundwater, you see quite a lot of variability, but not so much signal or impact of these new management practices. But if you look at just the wells that are less than 5 years old, you can see that actually there was an increase in the nitrate going into the water since the installation of these best management practices. And that was the switch from the manure to this inorganic fertilizer. that might be more easily leached into the groundwater, right? So yeah, so then yeah, and so if you look at the different wells, there was a lot of criticism of the study. Of course, nobody wants to hear like that they're contaminating everything, and so it's easy to pick holes actually, and it's not going to stand up in court. A lot of the work that you know the researchers do that are kind of more basic science, and so then they had to kind of refine the research question and get into a situation where they know exactly, okay, if we just look at these wells that are only surface, you know, they're only 5 years old, you can see there was some different trajectories. So like this well, this obviously switched from manure to fertilizer, whereas this one didn't switch at all and just used more more manure. And so they're all kind of going all over the place, and there's lots of variability. And so in order to better understand this kind of variability, you need more samples and you need more money, you know. So with the criticism, they were able to say, okay, we'll give us the right amount of money and then we can do the right study. And then that's what happened next. And so they took over 700 samples for over 5 years with higher frequency. Previously, they hadn't sampled in the winter when actually, you know, a lot of the manure was being stored and leached and accumulating and showing up later. And so, yeah, and so this allowed them to also target only this 5-year-old water instead of creating more noise in their data by looking at waters that actually didn't matter for the best management practices. And I forgot to say this is I think it's not a karst system. But it's mostly sand, and you know, it's very high flow rates through the. It's not much retention of the water, so you have to go quite deep in order to get this kind of anoxic systems where you would have denitrification. So that's why they're also looking at this younger water. Yeah. So. Then you get a lot more data and you start to see more trends. And so this is what they call a Keeling plot, and it's one way to analyze the isotope data by comparing it to the inverse of the concentration. And the notion is that if it's linear, then it's just showing you some kind of mixing of the two different waters. So basically you see intense mixing here of waters that are contaminated with manure with waters that haven't been contaminated with manure but are somehow more, they have this natural signal for the 15N. And then basically this is some sort of combination of different fertilizers showing up here. So they could already identify just 3 groups just looking at the simple, making a plot in Excel and saying, okay, manure, natural, and some combination, right? And then when you have this kind of data, you can also do more sophisticated modeling approaches. And so this, especially if you've defined the N members, then you can generate plots like this and using Bayesian clustering. And so there's a lot of stable isotope packages in R that you can basically input your data and it can do this kind of stuff for you. So the field is really quite advanced in assigning the inputs of different sources for nitrogen isotopes. And so then using this approach, you could see that there's this like 5 different groups that you could be pulled out for that could be assigned based on their nitrogen 15 isotope composition and the nitrogen content in the water. And so when you do this and you assign, now you can go to the map and take a well and say, okay, this well was among this group. And here they've defined 4 groups. The first 2 are kind of more natural signals. And so you can say, okay, these farms here seem to be complying with regulations. And some of them have some spikes in fertilizer, so most of the year it's fine, and then I think before they do some planting, or actually I think it was they add some primer to the soil so it maintains its nitrogen and carbon content. So they add a little bit of manure, and then it spikes every now and then. So you can say, okay, that's what's happening in these regions. Then there was another group, this green group that, yeah, so that's these, and then you have this mixture group here. Wait, and so they could basically, based on this modeling approach, they could assign these different wells to the practice, the fertilization practice that they had been using over the last 5 years. And so you have these different trends that they could look at as well. So if they look, this was collected over 5 years, and basically these are the different groups. And so what they're saying is this is the natural group. This is the one, a natural group that has also very high variability, and this is due to the spiking. So then this is the one that has been increasing. So this is, they have heavy manure loading, and so there's also no denitrification happening. So you know that the increasing signal in the 15N is not from denitrification, but from the nitrogen source. And then this one that was decreasing, and this was basically the switch. So they had high nitrogen contamination, but they had a decreasing trend. So they basically just switched from one to the other, and then they could identify which wells were showing which kind of transition over this period, and then use this to inform the policymakers. So one of the hard things to do with the policy is enforcement, right? So you have to have some kind of reports or letters, but if you can go in and say, look, this well has shown that that there's been an improvement, or there has been no change or something like that, then I think this is what people want to hear, on who aren't in the scientific discussion but are on the other side of the table. And so I'm going to close now with 2 slides, because one of the things that we think about a lot is how do we apply this approach to different systems? And we would like to suggest some sort of approach, like a schematic to make choices along the way of how often you're going to sample, how many sites are you going to sample, what do I need to know before I sample. And so you need to look at the different sources. Is it used for agriculture or not? You have to know the hydrology of the system. Is there a karst or not? Can you quantify the flow rates and the discharge and all of these things? And then look at the different types of land use. So the different fertilizers that are used by the by farmers in the region, or just how is the land being used that is influencing these wells. And I show another schematic now too, which is basically like a flow chart. So do you have data? Is it good? Yeah, everything is good. Yeah, keep up the good work. If not, then you need to develop a sampling strategy. Okay, we need to measure if there's like riverine discharge. And so this is kind of a flow chart that was developed by Mathieu Sabilo and colleagues, where they had kind of a meeting, and with the goal of producing something like this. And I can share this with anyone who's interested. And basically the different kinds of measurements you need to make. So conductivity, pH, dissolved oxygen measurements, together with nitrate concentration, which we definitely need to know in order to make nitrate isotope composition. And then if you have the other N species as well, it also is very informative of the different kinds of practices or biological processes that are happening. And then it also includes sampling recommendations, if your sample needs to be filtered or frozen or preserved somehow. So And then when we measure the isotopes, we also measure water isotopes, because this tells us a lot more about the hydrology of the system and if there's some impact from mixing or yeah, just the different inputs of water. And right, so with that, I would just like to acknowledge some of the funding sources and the groups who have contributed to the data that you've seen today, and definitely Len Wassenaar for all of his activity in designing this method and this study that I showed from him. So with that, I can close and take any questions. Thanks for your attention. How am I on time?
Primero en español, vamos a abrir un espacio para preguntas y respuestas, si gustan. Y también en redes, si hay en Zoom, Facebook. In English, next, we will open a space for questions here in the auditorium or en Facebook and Zoom. Any questions? ¿Preguntas por aquí?
Well, thank you for your presentation. I have two questions, actually. Maybe it's a little bit technical, the second. First of all, do you only take surface samples or do you also take water samples at different depths in the lakes? And the second question is about, well, you're measuring the nitrous oxide by IRMS and laser spectroscopy, but you're reporting the results as nitrates. So do you run experiments to know the isotopic fractionation factor, or do you use a standardized factor to know the conversion?
Yeah, thank you. Good question. So the first one's easier to answer. The Plesna Lake, it has routine sampling at the surface and I think 20 meters or something like that. And actually in the summer it's stratified, so it gets anoxic in the bottom. But this, yeah, this I haven't talked about that signal so much in this. This is more important for for the denitrification that might be happening, but we didn't see that so much in that system. In the other lake, I think they're only sampling the surface, and so we haven't seen anything about that. And in the other study, I showed they take wells, that I guess they have wells that are tapped to some depth, and they just collect it, and then they know how old the water is. And so for the second question, it's a very good question. So. I don't know if I'll go back to the slide, but if you'll remember, I showed you the different techniques that are being used now for measuring nitrate isotopes. And so all of them are converting nitrate to N2O because you need a gas to measure on the IRMS. And the denitrifier method is much preferred by places like USGS and that's the method that they've adopted because one, it works better if you have salt in your sample and like if it's getting like any kind of marine influence. If you don't have to rely on the chemistry, then there's like these matrix effects kind of go away and the bugs that are eating nitrate, they just eat all of it and so that's important because if you have 100% conversion from nitrate to N2O, then you don't have to worry about fractionation. It's 100%. But for the titanium method, we know it's not 100%. And actually, it's quite variable. And they did a lot of work to figure out how much titanium to add so that you get the optimal conversion efficiency. And I think it was like maybe 80%, like 70 to 80%. So you know that there's fractionation involved. And the way that you get around that is you just measure the known standards. So you have to buy these reference materials with a known value, and you measure those, and then you also do some extra controls because if the peak height isn't exactly the same, then the conversion wasn't the same because one of the things with this method is you always start with the same amount of nitrate nitrogen in the bottle, and so you have this. And if your information was wrong, then the efficiency conversion might be wrong. And so there's a small window where you can do a correction, and that's these controls where you take a standard and you just add a little bit more titanium or a little bit less, and you can do this linearity correction. It's not linearity, but it's a combination of linearity and this inefficiency conversion, like so, this fractionation that's probably happening in the reaction. So it's and all of these corrections add error to your estimate, and so that's why the USGS prefers the denitrification method, because I'm okay with plus or minus 0.4 or even 0.8 per mil in some systems, especially if you want to tell atmospheric nitrate from soil. You know, it's 70 per mil difference. So if it's like 1 per mil error, I'm okay. But for them, they're the kind of scientists who get called into court and have to really have precise measurements and without any kind of wiggle room. So it depends on what your goal is in the end, but it's constrainable. So yeah. Does that answer your question? Yeah. Yeah. Hi.
Yes, well, thank you for your interesting and informative presentation. I would like to know about the boron. You say that additional isotope, what for? Yes.
Yeah. So if you remember, the box for manure was quite large, and so then you can answer the question which manure, and if it's coming from poultry or livestock, or if it's human waste, then you have different boron isotopes. And so there's maybe you cannot distinguish sometimes if it's like septic waste or fertilizer, manure fertilizer. And so then the boron is used for that. And there's other biological indicators too. You can look at like E. coli and all kinds of stuff. And so, but boron is I actually don't know a lab that's measuring boron isotopes, but I don't know. Do you? No, no. Yeah. So I haven't gotten to that point in my research, but it's informative of which kind of manure or. Yeah.
Okay. Okay. Thank you.
Well, thank you for the presentation and for the visit. I'm sure we will have a very interesting week in the lab.
Yeah. Okay.
And following Mariela's question regarding the depths of sampling, do you also sample the sediment, like the interface water sediment?
Yeah,
well, okay.
You mean the pore water, like, or?
Yeah, or just like the boundary between the water column and the sediment.
You mean like the bottom of the lake?
And if,
I'm thinking,
or if you do some nitrogen isotopes analysis in the sediment.
This has been done, but it's been very like specific projects where they've like taken a core or something like that, and it hasn't been really related to the group who is going every 3 weeks and collecting stuff. And so I think actually, in the institute where I'm working, they're mostly looking at the soils around, and there's not so much focus on the sediment. So I'd be surprised if nobody had done that, but I'm not aware of the data. Yeah, okay. I mean, this is what I would do in the previous postdoc was taking the sediment cores and looking at the sediment water interface, and yeah, but yeah.
And then another question, more technical. What would be the minimum concentration of nitrate in a sample in order to have a good
so for the titanium method, it's about so it's 0.1 ppm nitrate nitrogen, and or like I think that converts to 7 micromolar nitrate, which is quite high for natural systems, and in particular the ocean. So this titanium method, it's hard to recommend for studying marine samples because I think they're typically like 3 micromolar, so they're like not close to the limit of detection. But for the inland systems, and particularly the systems that we're interested in, we've had samples where it was like 500 ppm. So we just have to dilute the sample a lot in order to measure it. So, it's fine for that kind of approach and for the fresh waters. Yeah. So, yeah.
We have a question from Doctora Rocio via Zoom.
Hi, Arturo, and hi to everyone. Thank you very much for this very, very interesting talk. Can you hear me okay? Or
Yes.
perfect. Well, thank you very much for your talk. I have a question of ignorance, really. When you mentioned the isotopic composition regarding nitrous oxide and that you can know the source of the nitrate, implying, well, using stable isotope approaches. In nitrification, there is two arrows that say denitrification. And what it's about, you can estimate the denitrification rates derived, or meaning that you expect that nitrogen becomes denitrification.
I think, let me make sure I understand the question correctly. But yeah, it's something I forgot to mention when I showed the slide. And it's slide number 19, if the technical, yeah.
Yes, that one.
Yeah, so this is something I started to say later, and okay, maybe I go, did I show them? I'll just not project it. But you're talking about these arrows here. Anyway, okay, we'll fix it. Anyway, so those are basically trajectories. And so if you are collecting samples seasonally, then and you see your data falling along a kind of trajectory that has a slope between 1 and 0.5, then you can assume that denitrification is a dominant process in this system. And that's basically for interpreting your data. There's also other signals for nitrification that you can see, because it depends. You can also look at ammonia isotopes or nitrate isotopes, but there's something about the extent of the reaction and this kind of fractionation that you see. So I don't know, is that answering your question?
Yes, because my second question regarding this is this denitrification issue also includes NMX, endemo, and other end cycle processes, or just as far as we know, is this expected only for denitrification?
Yeah, there's quite some variability in the slope of that line, depending on if it's anammox or if it's denitrification that's happening in the marine system or that's happening in inland systems. But in general, if you want to take, so like for example, for nitrification, you're converting ammonia to nitrate, and then you have to add oxygen. So the oxygens, there's some, this is a controversial statement, but there should be 2 oxygens coming from water and 1 coming from molecular O2, and that gives you the isotope signal of oxygen and nitrate. And then when you're removing that, it's kind of regardless, but there's definitely some groups that are looking like position-specific oxygen in the molecule as you're making like N2 and N2O, so you can learn more about the actual process if it's anammox. So this is getting really quite detailed, and this is there's research groups working on that now. So this is but there is variability of the slope, and that's all I can say to that at the moment. Yeah.
Thank you very much, because indeed I think this is a new notion, but because as far as I know, denitrification was only the main losses source in any system, and we know that now anammox and others can take a great deal in the in the losses of nitrogen of the systems, no? And I think we are researching that, and we are knowing depending on each system.
Yeah. One thing I will say more to that is that coming from an oceanographer to a landlocked state and working with soil scientists, is that the oceanographers are actually figuring it out first because it's so much more homogeneous than the soil systems. And so you can just take a profile in the ocean and you can say, oh, there was denitrification and then there was anemones happening. And then they're the ones who are like discovering these isotope patterns for those different kinds of reactions, and then testing it as well as they can in the lab if there's some cultural representative. Yeah.
Well, thank you very much. Nice to meet you.
Yeah, thanks.
Any other question here in the audience or Facebook? If not, then we can proceed to the diploma by Dr. Juan Pérez Quesadas.
En español. No voy a leer en español, está en español. El Instituto de Geología de la Universidad Nacional Autónoma de México otorga, a través de su unidad de educación continua y distancia, la presente constancia al doctor Travis Menor por la presentación de su conferencia dentro del programa de seminarios institucionales titulado Nitrate, Isotopes and Cycling en Bohemian Forest Lakes, ¿no? Por mi raza hablará el espíritu, Ciudad Universitaria, 26 de febrero del 2024.
Muchas gracias. Yeah, thank you. Yeah, thanks, Juan. Thanks to all the organizers, and thanks again for your attention.
Thank you, Doctor. So with this is the closure of this seminary. Thanks a lot for being present, and thanks to all that shared the presentation with us via Zoom and Facebook. And well, have a nice day.